Thank you very much, Tae Seok, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, so thank Kate. This is my great honor and pleasure to be here again. This is my second time. A few years ago, I gave a talk. I hope, I giving kind of new update today. Actually, the multiple new update. Now I am a professor at J. Craig Venter Institute, the hardcore synthetic biology institute, including multiple colleagues over there. But today, I'm basically wanting to share something a little bit different from what my colleagues are doing. There's basically really hardcore synthetic cell, construction with the multiple collaborators, I believe, including Kate. Today, I'm, try to give some other aspect of, I call it engineering biology or synthetic biology. That is actually not using real hardcore synthetic cell, but basically using the living cell as a chassis. That's one thing I want to tell you. But to make my presentation more useful for many of you, basically I wanted to give some kind of list of tools you potentially could use in the future. That's the one thing I want to give you today. So basically I presenting 4 tools that potentially useful for you, and that's basically my talk today. And I know the format is basically you waiting until late. But if you have some question, I'm happy to entertain your question during the talk. So please raise your hand and then speak if you wanted to stop me if you are not understanding or something is not clear to you. Before I'm going forward, I wanted to basically claim or some conflict of interest. I found a company called Moonshot Bio. My last name is Moon. Why not? I can also start a company. Company name is also Moonshot Bio. We wanted to do something ambitious. This company basically dealing with 2 different aspects. One is the plastic upcycling. Unfortunately, I'm not going to talk about this, plastic upcycling topic today. The other one is basically probiotic engineering. I probably touched some of that, about this company. This is my current project, or a little bit more than 10 projects. I don't have time to cover all of that, so I'm basically pick some of that probably most related to synthetic cell community. This is our institute, Craig Venter Institute, overlooking Pacific Ocean. I'm always tease, Kate. Kate, beautiful area, Minnesota, but, cold in winter, not that much ocean around that. And this is my institute. I shamelessly, advertise, postdoc position. If you are interested in, beautiful sunset, Pacific Ocean, and also at the same time, wonderful science, technology development, please contact me. So let me start with my vision or dream when I started my position. At the time, Washington University in St. Louis, 2020, actually 2011, that is when I, giving a talk for my job talk, and then I got a job, started, my position 2012. And at that time I asked, what if we combine successfully genetic sensor and genetic circuit and actuator together in a human-defined way, manner? And if that is possible, we can make bacteria, of course, work for us, including multiple different applications, including, for example, tumor-killing bacteria, that bacteria only gather around tumor cell based on some signal from tumor that could be multiple tumor specific, molecule. But simply you can also make bacteria gather around only tumor cell because tumor has a very fast metabolism. That means the oxygen level is very low. And also at the same time, tumor generate a lot of lactate because of the fast metabolism, and they cannot completely metabolize the carbon all the way to carbon dioxide. During that process, lactate production is very high. And if you somehow develop sensor that detect uh, both oxygen level, but that's on low oxygen level and high lactate level, you can build some AND gate, and then we can combine these two things together, and then only when oxygen level is low, and of course the lactate level is high, your bacteria start to produce toxin. In that way, your bacteria only kill tumor cell, not the normal cell, because as you can imagine, if you have some family member, friend suffering from cancer, the most, challenging part of the tumor therapy is basically differentiating between tumor and normal cell. And I clearly know because my father, actually, I lost last year, he also suffered from this non-specific, treatment problem. So we wanted to develop sensor, but when I started my position, we basically focus on one specific chassis, and that is actually probiotic. I do believe many of you, probably taking yogurt, very often or regular basis. I do take yogurt almost every morning. But what I want to suggest here is not just using probiotic for health benefit. I wanted to engineer probiotic. In that way, that probiotic has some sensor. As I mentioned earlier, the sensor could potentially detect disease. At the same time, the sensor kind of trigger some output. The output, I call it the actuator earlier, but that output would be some molecule that cure some disease. For example, bowel disease, if that is detected by the probiotic cell, basically that sensor trigger the production of some molecule that potentially cure, or simply some generating some color that is basically a diagnostic kind of application. So that is the overall kind of vision, we had, when I started my position, and we start to produce or engineer some sensor. After, a few years later, that is around, I believe, 2016 or 17. Around that time, I found some paper that is very interesting to me or shocking to me. That is basically people start to talk about neurochemicals or neurotransmitters that actually found in our gut. And then one striking kind of, fact I heard at the time was one of the neurochemical, very famous one, serotonin. That serotonin basically control our happiness level, but that serotonin basically not produced by your brain cell, but mostly produced by our gut cell. The number is crazily high, more than 90%, 90% actually produced by our gut cell. And then after I heard those kind of interesting facts, and I realized we could potentially engineer those kind of sensor that specifically detect those kind of serotonin in our gut, and then potentially, produce some color molecule or fluorescence that could be interesting way to, basically diagnose, some serotonin related kind of problem. Or, if you make the probiotic sense serotonin level, and then if the serotonin level is too high, your bacteria start to produce enzyme, basically degrade serotonin. That means, if the serotonin level is too high, your probiotic basically produce enzyme that degrade the serotonin. In that way, serotonin level is go down because serotonin too high is not necessarily good. But if your serotonin level is too low, your bacteria start to produce enzyme that synthesize serotonin. In that way, your bacteria start to produce serotonin more. In that way, you could kind of maintain your serotonin level. I call it happiness level. Let's say that is 30 to 100. In that way, somehow, if the serotonin, produced by gut, that, we heard the circulate, in the, blood. And then unfortunately, serotonin does not cross the blood-brain barrier, but, there are some report the serotonin produced by the gut somehow interact with some receptor in the brain. In that way, potentially, affect our happiness or some other, our mood, etc. So that was the basic idea to develop the neurochemical sensor based on those kind of multiple facts, including, some enzyme also found in the bacteria that is actually, living in our gut. So, but the problem at the time was we found most of the neurochemical we wanted to detect the structurally very similar. That is actually I'm giving you here two examples. One of them is the PEA and the other is the TIRA. And then unfortunately, one difference is basically one molecule has one oxygen in the aromatic ring, but the other is actually no oxygen, and that's the only difference between these two. But however, the consequence or physiological function is very different. One of them is basically just related to food poisoning, but the other is, of course, the psychological disorders. And then, if our sensor in the clinic cannot differentiate between these two, and then you just have the food poisoning and then go to your doctor's office, your doctor may say you are crazy because of misdiagnosis. So that should be, prevented, of course. So we tried to develop some specific sensor for 2 years. Unfortunately, we couldn't reproduce none of the reported publications. So we decided to choose some other mechanism rather than the one already, reported in the literature. And then I can explain, how we developed that sensor. I believe you see my cursor. Yes? No? Okay.
Yes,
Assuming,
yes, we
okay.
can see your cursor.
Okay, so typically, people try to develop sensor based on some transcription factor, and because that is kind of easiest thing, you can do. But typically, the molecule you want it to detect, in that case, let's say, white molecule, that basically bind to the, transcription factor that change the conformation of protein, and then that trigger binding to the DNA, and then you could have some GFP or colored molecule generation that typically people kind of generate some biosensor. Unfortunately, in our case, our molecule is the amine that is in this side, that is actually our neurotransmitters. And then the real molecule we found based on our some experiment, without this TNA enzyme, we cannot detect this amine. That means the real molecule bind to this transcription factor is not amine, but aldehyde that is converted by the TNA enzyme. And luckily for us, this aldehyde very short-lived. that means unstable, but still, once that is converted to this aldehyde that bind to the transcription factor, and then we saw the concentration dependent increase of the output signal, in this case GFP, and then we didn't find any aldehyde in the diet or in the gut, and then we can say with this fast reaction, that is a TNA-mediated amine to aldehyde conversion, and then we can now claim this is a really amine sensor rather than aldehyde. And then luckily the reviewer and editor buy this argument, and then we publish this one in Cell System. But by that time, the problem was TNA converted kind of conversion, and also the transcription factor binding to the ligand. That was not specific at all. That means the all four compound actually giving the output. And we spent already 2 years, and this is the Navy, project, and then we only have 1 more year, and then we got hit by the COVID, and then we basically don't know what to do, except luckily we got 1 year extension automatically because of pandemic. And during that time, luckily, one of my students, Matt Amrafael, during the pandemic, we basically shut down for several months, nothing to do experimentally. But Matt somehow approached me saying, I want to learn this kind of, protein prediction algorithm that including AlphaFold or Rogera. And then I really delighted, because we don't have any other way to go, because at that time we basically done directed evolution to change multiple, random location, and then error-prone PCR-based kind of screening that require 10 to the 7 or 8 different cell, but we suffer from false positive or negative, keep failing to, find a very specific sensor. But interestingly, after he learned how to use that, algorithm or the AlphaFold or Rogera, and then he successfully identified 5 different residue, including 81 residue. And then we basically made all 20 variants for each residue, and then only screen 100 different location, 100 different variant by changing each 5 amino acid into 20 different one, and then we found multiple hit, including A81L variant that only giving signal when we use PEA molecule, but not other chemicals. So to make the long story short, I mean, this is the actually first demonstration for specific, neurochemical sensor, even without any crystal structure of VRR, that transcription factor we use, we able to use, this computational method to predict, and then actually the changing the specificity. And then after that, we wanted to expand that, effort by looking at all the intermediate to make this type of neurochemicals. And as you can see, this is a very complex kind of conversion network, but we found using same, AI or computational based, protein prediction algorithm, we identify individual residue, and then we done multiple of them, and then we found very specific sensor, for example, here, including tryptamine sensor that only, react to tryptamine, but not other, molecule, around the 10 different molecule. We didn't see any, response. And then we also found multiple different compound-specific sensor, including one of those interesting molecule that found in the gut at the same time in the soil, that is called auxin, indole 3 acetic acid. And we now start to use this oxygen sensor to understand plants and soil bacteria interaction or communication. And in that way, I mean, you could also understand gut, metabolite interaction at the same time, the soil plants somehow soil bacteria and plants kind of interaction using this sensor right now. Also, if you have any interest, I mean, those kind of things, always I'm happy to talk with you for potential collaboration. And then, more than 10 years of sensor development, we recently published one paper, the review paper, I believe longest paper I ever written, 20 page, more than 200 references. I believe this guidebook or the paper published in Cell System probably potentially help you to design and engineer the sensors. Okay, I've already spent 20 minutes each, and then I'll probably speed up a little bit more. So one thing, I want to mention, so far, we try to develop some sensor and then potentially some actuator that potentially make you happy or, disease detection, etc. But at the end of the day, this bacteria, potentially in, your yogurt, but bacteria is a live cell, and then that gonna be in your gut doing multiple function, including making you happy, detect disease, or curing disease. But that bacteria is unfortunately, living system that potentially going outside. And then that means, I mean, we need to worry about biocontainment. And then that is one thing I want to do. So since, I mean, we started this, probiotic project, we, start to consider this biocontainment, strategy, and then we try multiple different one, including, of course, the kill switch development. And then let me explain, how we develop this kind of kill switch. During this kind of more than 10 years of time frame, I mean, unfortunately, a lot of people also try to develop kill switch. We actually, scooped by multiple big name, Jim Collins group, Pam Silver's group, and also my own advisor, Chris Voigt lab. And they actually developed beautiful kill switch over the last 10 years. And then after we try multiple different things and then basically drop multiple project because of somebody already done, and we decide to choose a CRISPR system. And then interestingly, CRISPR system is actually a very good one, but very difficult to control because the CRISPR, if that is leaky, that is basically clip your genome and the bacteria die prematurely. But we able to fine tune the expression level of this CRISPR system without adding, of course, the homologous DNA. That's how typically you do when you use CRISPR for genome engineering. But for us, CRISPR is basically molecular seizure that clip bacterial genome. And then once bacterial genome is cut, basically bacteria, in theory, should die. And luckily, if you're working on the eukaryotic system typically have non-homologous end joining mechanism. That means even if eukaryotic genome basically clip, eukaryotic somehow survive with some scar in the genome, but most bacteria do not have non-homologous end joining mechanism. That means bacteria in theory should die. And the good thing about we actually chose Cas9. Cas9 could be generalizable because many bacteria actually potentially we could develop Cas9 based kill switch because Cas9 somehow working in many of the bacterial system also already reported by other group. So actually we decide to choose a Cas9 based kill switch development. I believe 2018 15 or something like that. I mean, many years ago, almost 10 years ago. But as I mentioned earlier, expression level tuning is not easy thing to do. And then to get this, very simple two graph, it took us more than 3 years. And then I really, appreciate Austin. So I mean, hard work without giving up. I mean, just, to make this, project working. I'll explain this data a little bit slowly because this is the key data, and y-axis is basically log colony forming unit. You can say the cell number per ml, and then x-axis is multiple different guide RNA targeting different genome location. Our initial hypothesis was if we have one copy of gene or 3 or 7. If we have the 7 copy, that means the more chance of cleavage. That means the more, efficient cleavage. That means more efficient killing. But it turns out, number of the cleavage site doesn't matter much because we see the relatively similar, cleavage activity, and we luckily showing 10 to the fourth reduction in terms of, cell number. Here, 0 means, I mean, no induction, 100 means the ATC, 100 nanogram per ml. That's the induction of the system. And then this is the, the most important data set. That's a control. Basically, control does not have any leakage or, the reaction in terms of the cleavage. So we are, happy with this data set, but unfortunately, we still have the survivor that is around 10 to the 4 cell per ml, and that is a high number compared to NIH guideline that is basically 10 to the -8 escape rate. That means if you start with the 10 to the 8 cell, at the end of the treatment, only one bacteria should survive. That's the acceptable level by NIH. So we try to figure out what's going on, and obviously the most important problem was the mutation. And as you can imagine, mutation is random and then very rare event, but we are basically selecting by killing a lot of bacteria. But as soon as one bacterium survive by mutating, like deleting this promoter region or some other region that basically destroy kill switch function, that one bacterium survive, and then that become 2, 2 become 4, and then multiply and then dominate entire population. So random rare event of mutation, it doesn't matter, even though that's random and rare, the amplification basically happening during this kind of population increase over time. And then that is kind of problematic. That means we need to achieve 100% complete elimination of bacteria, and then actually we stopped by that time. But we already spent 3 years, and then we realized what kind of thing we can do. So we try multiple different things, including we actually knocked out error-prone polymerase that already exists in the genome, knocking out that gene copy, error-prone polymerase. We improved the mutation rate a little bit better. And we done multiple different one, but that doesn't necessarily kind of solve the problem. And then one day I was sitting in the hospital waiting for my, doctor's, office appointment. But as you can imagine, in United States, medical system already been not efficient. Even if you make an appointment, you need to sometimes wait 2 hours. And at the time, I didn't bring my computer or cell phone, and I start to daydream. And then at that time, I just asked myself what happened today if we lose power. And luckily, in most developed country, if the power just gone, we always have the backup generator. In that way, hospitals still in power, and then doctor continue to do any operation because that is kind of safety measure. And then I realized, why not? We could have multiple kill switch. That means even if one kill switch is not functional because of mutation, and then the random mutation, may not happen multiple different location if we change kill switch, sequence. In that way, you could have the multiple different kill switch in different genome location. And then we basically try to figure out how to build kill switch in the genome because we basically put Cas9 system into the genome. Making multiple kill switch sounds trivial, but in practical, practically it's not easy because we using Cas9 to kill the bacteria at the same time use Cas9 for genome integration like you do for the genome engineering. But Austin still figure out how to integrate multiple kill switch in the genome, different location with a different sequence of the promoter. And then we put up to 3, and then we saw complete elimination in vitro when we use, flask culture. And then to make sure we actually decide to add one more. So we now have the 4 Cas9 in the genome. And then we basically use mouse model, and this is basically data I'm showing right now. In the mouse model, we basically showing here controls stay in terms of numbers stay the same, even multiple days, but the kill switch probiotic basically completely eliminate by the time 48 hours, and then we see no bacteria survive. And then this is actually when we use ATC as a molecule because some people, including some NIH reviewers or even NIH and FDA, worry about probiotics stay in the gut for a long time, somehow become pathogenic. At that time, we wanted to use this kind of system to kill only the bacteria, our GMO bacteria, rather than affecting other bacteria, including the beneficial one in the gut. Because if you use antibiotics, all bacteria die, and then that triggers some other problem, including bowel disease. But we demonstrate by using this technology, now we can only kill one bacterium that is a GMO we created. But real biocontainment is not this way because you don't want bacteria, as I mentioned earlier, that's a live cell, and then the bacteria basically go outside. I'm sorry, I'm talking about the poop or fecal matter. With the fecal matter, bacteria go outside, and you don't want to spray ATC just following your poop or the waste treatment facility to just kill the GMO. So we decided to use some other sensor rather than ATC, and then we realized temperature sensor might be a good one. And as you can imagine, our body temperature is always 37, and then we developed very, interesting temperature sensor that actually on at 37 degree, and then completely off at lower, Celsius. And then, as you can imagine, this is our body temperature. And then typically outside of our the body is lower than 37. Even in Africa, at least one day or one night out of 7 nights. And then in that way, if the bacteria face lower temperature, we can make the so-called inverter. In that way, kill switch only working at 30 degree. And that means the bacteria basically die outside of your body. So that is what we done. And then we use the same, mouse model, and then we demonstrate bacteria really die at 30 degree, but not 37. So that is the technology we develop, or the tool we develop. Just kill one GMO, we target it without using antibiotics, and then that is actually success. But the Austin also come up with new idea, and that's one thing I wanted to develop for many years. That is basically the tool to engineer microbiota or micro community, very specific, targeted way. Let me explain what we wanted to try at the time. As you can imagine, genomics advance by using knockout tool to understand the function of individual gene in one single cell. We now understand entire E. coli's genes function because we knock out one by one, understand, oh, this is the alcohol dehydrogenase, basically converting alcohol into the aldehyde. But once you knock out this gene, there's no such reaction. In that way, we know the function of that gene. So we now understand entire, E. coli cell. We also understand, many synthetic cells kind of function, including Kreppventer's, cell. But do we have same level of understanding in terms of billion different bacteria community? The answer is no. We don't have any, understanding of the individual bacterium's role, except some well-known bacteria, let's say fixing nitrogen or doing some photosynthetic, carbon capture, etc. And then we try to develop similar tool to engineer microbiota, in a single strain level. But as I, explained earlier, CRISPR might be a good, tool to do so. And then, but problem in terms of this kind of engineering is we are not talking about, in theory, billion different bacteria community and then only target one bacterium killing. This is not easy problem to solve in terms of even guide RNA design, because you need to have guide RNA that's uniquely bind to the your bacterium of interest without binding to other, bacterium non-specifically, and how you're gonna design this one manually. So Austin come up with some algorithm that basically scanning entire, system or, billion different bacteria. And then this algorithm basically scan all the genome sequence and then identify one specific guide RNA that only target, our specific, bacterium of interest. So that is one thing, he come up with, and then we developed that algorithm by using machine learning. And then even that algorithm spit out guide RNA that potentially high cleavage efficiency based on all the data over there. So now, you don't need to worry about how to design guide RNA as long as you know the sequence of your community or the identity of your community, and then you can just input that one, and then your, our, program basically spit out this kind of guide RNA sequence for you. But second question we need to ask is how to deliver this CRISPR system into the community. And he basically, come up with another, method that is basically using liposome. I don't believe, I need to explain what liposome is for this community, because many of you using liposome to make synthetic cell or synthetic membrane. But for us, liposome is basically delivery vehicle that basically, lipid bilayer that basically, we basically adding to the community. That could be your gut or soil. And that liposome basically only contain some DNA, not entire genome, but the DNA encoding guide RNA. And of course, the CRISPR Cas9 gene. And then we, at that time when we try, we kind of worry about efficiency, but interestingly, this lipid bilayer, because that in hydrophobic nature, that is non-specifically fused with any bacterium in theory in the community, and then deliver DNA very efficiently. That's what we found. Of course, not 100% efficiency, but still kind of, significant delivery into the cell. And as I mentioned earlier, specificity now comes from guide RNA. And then even if some other bacteria, for example, DH10B in this graph, or MG1655, that's a very, sequence-wise very similar, but only this cell that is actually die, but other cells survive using this method. So we basically using the same technology to also basically isolate only one bacterium out of billions of different bacteria by using homologous DNA, basically, recombine that homologous DNA into the genome, but that, homologous, DNA contain antibiotic resistant gene. In that way, bacteria basically take that, antibiotic resistant gene, and then we screen using antibiotics, and then bacteria only survive when we have the specific, guide RNA that is targeted engineering, and then we basically showing only Nisser cells survive, and then all other bacteria die. And then we actually use that one using fecal matter. There's some multiple different bacteria, billions different bacteria over there. When we spike our bacterium, only our bacterium die, or only our bacterium isolated in this complex real world kind of sample. So that is the actually, Austin's real achievement, and he kind of published multiple big paper out of this kind of his PhD, and then I really respect his kind of persistence for more than I think 60 years. I mean, but publishing multiple great paper out of this lab. One last thing I want to mention is I call it ARGFP. That is the antibiotic resistance gene-free plasmid system. I do believe many of you made plasmid in your life. And I believe once you make the plasmid, and then you basically autoclave or make an adding bleach to kill the bacteria. But one thing I want to ask, how many of you destroy DNA? And probably the answer is none of them, because autoclaving or adding bleach, we found DNA is still intact. And then there's a current, we currently, writing a paper about that kind of result. That means most of the plasmid survive. And then when you dump or dispose this dead cell, because that is a current practice or regulation, that discard or dispose dead cells still contain plasmid that release into the environment, and that typically contain antibiotic resistance gene, and then that potentially contribute to, of course, antibiotic resistance spread. So we want to solve this problem by developing new cloning method that does not contain any antibiotic resistant gene. And then what we done is, because of time, to make the entire process very simple, we basically knock out auxotrophic gene and also essential gene from the genome. But before doing so, we basically clone one bacterium plasmid that basically contain, of course, the auxotrophic gene and essential gene. But this plasmid basically contain temperature-sensitive origin, or sucrose-sensitive origin, and this intermediate plasmid transform first, and then knock out the genome copy. And then if you do whatever you're cloning by using Golden Gate assembly, Gibson assembly, once you do a transformation, And then, basically screen at the 42 Celsius for temperature origin, curing, or sucrose media, and our intermediate plasmid just gone, and then only your plasmid of interest basically remain. In that way, we achieve the plasmid, cloning method that is actually ARG free. And then now we put that into the adagene, both plasmid and strain. The only thing you need to do is just buy this one by paying some small fee from adagene and then do the same cloning method. The only difference is don't use any antibiotics. That means, you don't have any guilty feeling anymore by using antibiotic resistant gene, in your plasmid and potentially release into the environment. And then your PI potentially save some money by not buying the antibiotics. So we kind of demonstrate, again, using mouse model, our, plasmid system stay, almost 100% without losing, for 12 days. And then in vitro, more than 1 month, we kind of show this, plasmid maintain. With probably around 2 more minutes, I want to advertise my seminar series. Kate did phenomenal job, over the years, doing the synthetic cell seminar series. I also do have one seminar series called Synthetic Biology Young Speaker Series. And the focus is a little bit different. I invite 2 people every week. One is the pioneer, and the other is the, I call it rising star. Kate was one of the rising star, beautifully, gave a talk, many years ago. And then in that way, I kind of basically ask some pioneer deliver their vision or some, advice to young people. And then this one actually followed by 45-minute talk by the young speaker. And that is actually the main talk of this seminar. In that way, nurture a young speaker. So if you are interested in giving a talk as a pioneer only having 5 minutes, let me know. I'm happy to schedule your talk for next year because I've already scheduled entire year this year. If you are young speaker also wanting to give a talk, please send me CV and then the committee will consider your talk nomination. And then we have multiple, speakers so far, and then we realized we were going to meet together, and then we actually had the first in-person conference in Hawaii in December last year. And I believe people in Minnesota want to spend some week in Hawaii, especially in December. So we have multiple people over there, but currently the webinar speaker include 1 Nobel Prize winner, 24 National Academy members, 42 funding agency director, and 20 synthetic biology journal editors-in-chief, and 11 Nature and Cell journal editors, and of course, the most importantly, almost 200 rising stars line up since 2021 all the way to actually next year. So if you're interested in giving a talk again, please contact me by sending email, CV attached. And this is the one paper I wanted to also let you know. If your life is miserable because your paper never accepted or your experiment never working, I guarantee by reading this one-page paper, you may already feel better. That's one thing I want to mention. And I'm also chief editor of New Biotechnology and 9 more journals editor. And if you're interested in submitting some paper, and just send me cover letter, and then I'm happy to entertain your submission. With that, I want to thank all my student and postdoc, and also my 2 advisors still giving me advice, Chris and Chris. Now at MIT. Both of them is the department chair and also funding agency. And also I'm happy to, I'm also thank you for your attention. I'm happy to answer any question you may have. Thank you.
Thank you. Thank you so much, Tae Seok. That was amazing. There's a lot of questions in chat. Can you see chat?
Let me go. Okay.
If you can read the question out loud, because the recording doesn't show chat.
Sure. Okay, so I have happy face, the first one. Okay, thank you. And Ting who asked question, sensor work is very impressive. Could those sensors be used for a long period of time, like deployed in a gut and work for a month? Fantastic question. For the sensor, we plan to test uh in the gut. And luckily, or interesting thing, we heard from the NIH and FDA, they do not want probiotics stay for a month because of the safety reason, because somehow public have some, impression this GMO potentially become pathogenic in the gut, and that, having some probiotics stay for a long time, that's not a good idea. That's typically the regulatory agencies kind of concept or public concept. So they want us to basically have probiotic not colonized in the gut. So that's why we haven't tested for a long time. The only thing we tested is only for, I believe, around 12 days or 2 weeks. And during that time, I would say some basic sensor still functional. But we haven't tested like the more than 2 weeks. That's the one thing I want to say. But we currently have one, probiotic that contains some sensor, actually currently using some mouse model to test the longer kind of period. But the other thing I want to mention is not in the gut, but we demonstrate already using kill switch. That kill switch actually in the in vitro, that means in the flask, that is actually stable for 4 weeks without any problem. But in the gut, and we only tested probably around only 2 weeks. And I believe that probably answered the question. And second question is Emmanuel asked question. This might be naive question. There is no naive question. Everything is great question. But why kill switch cannot be a bacteria missing an essential gene, something, for example, to make a vitamin? Fantastic. So a lot of people already develop oxotrophic, biocontainment. But the problem of this method would be those kind of compounds also do exist in the environment, and then so-called cross-feeding, basically making them survive in the environment. That's why we, basically try to develop kill switch, even though kill switch development is much, much harder and challenging because of the mutation problem. Also, mutation problem also applicable to oxotrophic method because, somehow mutation, make bacteria gain that function, let's say vitamin production, that, is not also a good way to run. So that's why we develop kill switch, and that's the actually basic answer. Yeah, so that is actually the last question is also the same thing. Emmanuel believe, those kind of oxotrophic method cannot be escaped, but there are multiple ways to escape, including, cross-feeding and mutation, etc. That is actually my answer. So that another question from Adam. I think I don't know what this. Probably this is a typo. Okay, so that is actually a question I now realize. And Ben asked a question, Thanks so much for your presentation, Dr. Moon. It is really interesting since I'm currently working on phenylalanine tyrosine dual sensor. Okay, fantastic. Actually, we published one paper in the same cell system paper that already developed phenylalanine sensor. I highly recommend you check out. In that way, I don't scoop you. I think that the temperature sensor is really interesting since it is parameter, I don't know, parameter well regulated for mammals. Yes, the temperature sensor potentially working, but the one caveat is our temperature sensor coming from bacteria. So that means the mammalian cell, that should not work because the promoter system, everything should be changed. I want to ask if you test the kill switch in a higher temperature like a fever or infection environment. That's a fantastic question. Actually, our sensor originally coming from pathogenic bacteria, and then the original temperature sensor actually transition around between 40 degree, that's actually fever inducible sensor. But to use for our goal, actually we basically engineer by mutating, I believe, 3 residue of amino acid to make the transition temperature a little bit lower. That's now, I believe, 34 or 35, but we have the sensor that's originally from nature that actually trigger at 40 degree. And other group, I believe the Shapiro group at Caltech actually using that sensor to kind of fever trigger kind of action. And then, but we haven't tested that, but other group already tested the same origin in a temperature sensor to test for fever reaction. And the second question is so what we could spec the system in this temperature? What is spec? I probably speak of this system. Okay, that is probably. Kate, do you know what is in the question, Ben, or could you clarify what is question?
If Ben is still on call, yeah, I see him. If you want to unmute yourself and clarify the question.
Sure. Thanks so much, Dr. Moon, Kate. I just want to ask it what we are expecting for the behavior of the sensor in higher temperatures or in fever environments.
Yeah, we have as I mentioned earlier, so we have the sensor that changing around 40 degrees. The transition temperature is 40. That means 37 do nothing. But at 42, basically this or 40 or 42, that temperature sensor actually trigger, and then we can use that one for fever detecting, etc. Yes, absolutely. That's original pathogenic sensor, because pathogen wanted to attack us at high temperature in fever. In that way, basically infect together. And that's actually, they use that kind of temperature sensor, but we basically use that same sensor, but by mutating residues. In that way, basically transition at the 34. In that way, we use for our goal, because we want to have 37, not nothing, but 30 degree basically trigger the whatever kill switch. I believe I cover all the question in the chat.
I think you did, yes. Thank you so much. No, I think that was just a comment. Yeah.
Sure.
Thanks so much. That was really inspiring. I'm really curious to see if we can use some of those temperature sensor circuits in cell-free systems. That was kind of my.
Happy to share the philosophy for you.
Yeah. I don't see any other questions, and I want to say thank you again, Tae Seok, and thanks everyone else. We're past time, but it was great. And
sure.
Have a great rest of your week.
Okay, see you somewhere in the conference.
See you soon. Bye.